Great. And I can take this off, right? Great. Thank you so much. And thanks to Discovery on Target just for organizing an in-person meeting, still hybrid, but it's great to be able to talk to people at the meeting. So it's a pleasure then to tell you today about our next-gen kinase and phosphatase drug development and the use of PhosphoSens for activity-based detection of the activity of these important classes of enzyme. So I don't have to tell this group that protein kinases and phosphatases are a hot topic. So we've really seen an amazing surge in the activity, and it's really driven by two major drivers. So one being obviously biopharmaceutical industry's pursuit of blockbuster drugs. And so roughly 30% of all drugs approved by the FDA over the last 3 years then have been protein kinases, and almost 40% of the value of pharma deals, pharma biotech deals in the last decade have been around protein kinase inhibitors. So it's a very large market with a major focus on oncology, but we're seeing more and more really novel inhibitors being assigned or developed for inflammation, for neurodegeneration, and other disease areas. The other important area is the opportunity for disease diagnosis and the demand then for personalized medicine. And so having tools then that could be used as biomarkers for disease, to predict disease, to be able to understand drug efficacy, and to be able to monitor resistance. So we see a lot of activity then in this area, major recent fundraisings from multiple biotechs that have been started, lots of acquisitions. So just a really very healthy time right now to be looking at protein kinases and phosphatases. And if we look at the number of inhibitors then that have been approved by the FDA, still none for phosphatases, but kinases obviously are very strong. So a really nice trend, but if you think about the billions of dollars that have been invested to be able to do this, right, it's really not a very efficient process. So we really need to improve this and improve it across then different disease areas. And so there are many reasons why it's been very difficult. And so really legacy kinase tools have limited patient and shareholder value. So despite the huge market size, what's available is inefficient, costly, or both. So the current drug pipeline then for kinase-targeted therapies is severely handicapped. And you see this in terms of the average cost to develop a drug being $2.6 billion, and only 3.4% of clinical trials actually succeed. And even then, what you see is the efficacy can be rather low. So drug discovery then is constrained both in scale and speed, and really is due to the lack of suitable tools for effective development then of new kinase drugs. And lastly, only 15% of the kinome, or 518 protein kinases, have been explored in terms of developing therapeutic leads, and there are no phosphatase-approved drugs by the FDA. And in these cases, then, assay limitations are a major factor. So what do we need? We need better tools, then, to accelerate drug development, reduce costs, and improve the efficacy of those drugs that are developed. These new tools must allow researchers to identify promising and lead compounds from thousands to millions of drug candidates. You have to be able to conduct specific measurements that predict drug efficacy, and you have to enable rigorous testing at all stages of the drug development process. And so now we see this as a very fragmented assembly of groups, assays, and so forth that is relatively inefficient, and because there's really no platforms that work across the entire drug development workflow. So, of course, where there are problems, there are opportunities, and so whoever solves these major kinase assay issues will own the market, we hope, and while creating value for drug developers and patients, really important. And so we believe that PhosphoSens then by AssayQuant is that game-changing platform for next-gen kinase inhibitor drug development. Again, focus on kinases, but I'll tell you what we're doing with phosphatases as well, where we're seeing a lot more interest in that target class. So it's really, with that vision that we founded AssayQuant, and so AssayQuant was founded by both myself and Dr. Barbara Imperiali. I have 25 years of experience in the life science tool market, developing a variety of assay platforms, really bringing to market then the ability to select and develop more effective inhibitors. Barbara is the inventor of the PhosphoSens technology, actively involved in the company. She is an MIT faculty member in both the departments of chemistry and biology. She is a National Academy of Science member and just a consummate inventor. So it was really a great pairing when we came together to be able to create the company. And the first flagship then product line that we've created is what we call PhosphoSens. And it allows detection in two different formats. So you can use these sensors for continuous kinetic detection of kinase activity. And so what's shown here is you take a peptide substrate, could be any size, can even be small protein substrates. You synthesize those with the SOX fluorophore shown here, and it's put in proximity to the serine threonine or tyrosine residue that is phosphorylated. You take this sensor then, mix it with kinase, ATP, and magnesium, you get phosphorylation, and you get immediate then generation of a fluorescence signal, anywhere from 2 to 10-fold increase in fluorescence, which is really based on the affinity for magnesium. So the non-phosphopeptide has low affinity for magnesium. The phosphopeptide has very high affinity for magnesium, but you can use physiological levels of magnesium in your assays. So what you get then is an immediate then progress curve that shows the initial linear region, which allows you to calculate the reaction rate for a particular reaction with or without drug, with or without any sort of stimulus that you want to use to modify the enzyme of interest. So it's all based on chelation-enhanced fluorescence using the SOX chromophore. SOX is covalently attached to the cysteine residue, so it doesn't come off. You can use reducing agent. And importantly, you can have any amount of flanking sequence on either side of the phosphorylation site. So what I'll show you is that we can make really great sensors that are based on physiological sequences. SOX is small and minimally hydrophobic, about the size of tryptophan, and so it avoids artifacts seen with larger, much more hydrophobic dyes. The assay is direct, homogeneous, and kinetic. So continuous, so it requires no time points. It's an add and read. You walk away and do something else, and then come back and collect the data and do your data analysis. So it's ideal for quantitative assessments, including profiling, potency, and mechanism of action determinations, and covered by multiple patents, and we have the exclusive license then from MIT. Importantly, so a lot of groups came to us and said, we love the SOX fluorophore, we love the kinetic format, we really would like a redshifted version. So can you make us a redshifted SOX? And if you look at the redshifted dyes that are commonly used, you know, Alexa 647 and others, I mean, they're really large, right? So they're grease balls. And so you get a lot of artifacts with hydrophobic drug compounds. But it turns out with PhosphoSens, it's actually quite simple. So the SOX fluorophore can serve as a chelate for europium. So at any time during your kinetic reaction, you can add europium. That then allows the excitation to then sensitize the europium luminescence, and you get a signal at 620 that is a time-resolved fluorescence assay. Okay, so redshifted, you can use 100 microsecond delay, so you eliminate any sort of interference with compounds. So now you can see, you can go back and forth from an HTS mode to a kinetic mode in a fit-for-purpose manner. So it uses the same optimized, CSox peptide substrates, but in an endpoint format. SOX serves to sensitize the europium luminescence. Again, you get the 620 readout. Ideal then for compounds where you see interference. And this is the workflow. So really simple, right? It's an add and read assay, whether you're doing kinetic or endpoint. You can do this in any plate format. We have many groups who have miniaturized this down to 4 microliters in 1536 well plates. You can read it on any reader. there's a whole series of readers that are really good. We use the Biotech Synergy Neo 2. Again, add and read. And then you could determine, for example, use kinetics to be able to look at enzyme kinetics, drug properties, enzymology, or you can use the endpoint read to look at drug screening, SAR, and so forth. And we are increasingly developing more selective sensors that can be used with crude cell lysates, and I'll touch on that at the end. So an important part of the technology was being able to make high-throughput CSox peptides. And so we have 30,000 sensors that we've screened, a variety of different branches of the kinome so that we have physiologically relevant sequences that have been optimized for, various targets in the kinome. And so we can put CSox at -5 to +5 and be able to then understand where is the best location. And this just gives us then optimized peptide sensors. And we've incorporated this into an optimization process where the first milestone for assay development is to screen subsets of these 30,000 sensors. The second milestone, we can take the best sequences and we can do individual amino acid substitutions at each position and determine which best helps the peptide be presented to the kinase. And in the third milestone, we look at all the favorable combinations so that we really identify where the presentation of the CSox sensor is optimized for a particular target. And this works really well. So we can go in and look at a certain branch of the kinome, in this case, tyrosine kinases, and we can select then for sensors that are really generic, that see 80 to 90 tyrosine kinases. We also have some really generic serine-threonine sensors. But importantly, we can also pull out sensors that are really selective for your target of interest. In this case, a single peptide that is very selective for KDR or the VEGF receptor 2. So this process is just outlined here, where if you look at the round 1 on the far left, this is a peptide where we've done now milestone 2. So we've substituted individual positions with 16 different amino acids. And every progress curve is a different peptide that differs only by 1 amino acid. And so you can see there's a dramatic effect in terms of the ability to phosphorylate that particular peptide. If we look at the combinations as shown in round 2, you get some that don't work really well at all, and you get some that are really synergistic in terms of the substitutions and the favorability of that final sensor. And we can then select for sensors that work across a particular target, like EGF receptor, to not only see the wild type very robustly, but we can also detect panels of mutants so that we can use the same sensor to interrogate drugs, for example, for the EGF receptor. And we've done this then for many, many targets, and I'm just highlighting here a variety of difficult targets where pharma customers came to us and said, we really need an assay that is much more robust. Our bottom of our assay has fallen out, and we want something that's much more drug development optimized. And so these then are all examples where proteins are typically used as the substrate for these kinases, and we're able to select for really optimized peptide-based CSox sensors, thereby enabling all of those assays. So we can use the same approach for phosphatases. We start with a CSox-based phosphopeptide sensor. You then add phosphatase, and you're looking for the dephosphorylation or loss of signal. Again, you see, really nice quantitation. Same mechanism of chelation-enhanced fluorescence. You can use both kinetic or the endpoint read format. And again, we just use physiologically relevant phosphopeptide substrates, where we actually see really nice selectivity between these phosphatase assays, so we can develop different sensors that would allow substrate-dependent inhibitors to be developed. And just some examples showing here with PTP1B screened with the AQT 266 sensor. You can see on the left, you add increasing amounts of phosphatase, you see rapid dephosphorylation of the sensor and loss of activity. You see really nice linear linearity in this system down to very low picomolar levels of phosphatase. And in the lower right bottom is just showing you that you can use then the PhosphoSens red with europium. You see a 7-to-1 signal to background Z primes over 0.9. So the whole system gives you really high Z prime values in kinetic because it's a reaction rate, in the red because you get really good signal to background ratios. So we've expanded this whole approach to other target classes. So we've just developed a whole series of assays using the PhosphoSens technology, using the SOX fluorophore to detect the activity of diacylglycerol kinases. We have about 8 assays right now working on the last 2. So there are 10 isoforms altogether, and they really differ a lot. So both in the optimized conditions, and you can see their subdomain organization. So in this case, we're not using a peptide. We're actually looking at phosphorylation of diacylglycerol to generate phosphatidic acid, and that's what we can detect in the PhosphoSens platform using a modification of the SOX fluorophore. So these assays then are very popular. We're seeing here on the left is looking at different DAG species. So there's about 30 different DAG species that one can use, and you'll see specificity for individual isoforms. And so here is just looking at a series of DAGs, looking at the progress curve over time, where you see phosphorylation of the DAG in real time to create phosphatidic acid. And we can then select for a particular DAG species that's optimum for a given isoform. In the middle then is just looking at enzyme linearity, so increasing amounts of DGK alpha, and you can see it going down to really low nanomolar levels. And on the right, looking then at a particular compound that allows us to see really potent inhibition then of DGK alpha, and we can run screens where we look at all 8 of the DGK isoforms to ask whether there's specificity in terms of those individual drug leads. And importantly to mention that all of these assays are done in a detergent lipid micelle, where you create a complex where the DGK, the DAG, and the lipid and detergent, again, form a micelle that allows you to get selective signaling. So a lot of advantages of our platform, and we've had many customers who came to us and said, we really love the PhosphoSens platform. Why don't you create a profiling service? Because this is something that is a really critical need. So if you look at the advantages of our assay system, we have a single-step process. It's a real-time analysis. You can use any ATP concentration. You can use complex samples. We can price it to be very cost-effective, and it works across the entire workflow. So it really gives us really strong features and benefits that distinguish it from any other platform that's currently offering profiling service. And so we've done this. We've actually now put together 372 assays in our profiling service, as shown here in a kinome view. And the results are really dramatic. So clients are saying that it correlates with their cell-based assays in a way that no other platform has. And there are many reasons why. So we can do this at high ATP. So most assays use low ATP. But the body has millimolar, so we're going to run our assays at millimolar ATP. We run it kinetically, so we understand during the whole profiling reaction what's happening with the enzyme. And so we get initial reaction rates that are much more accurate in terms of assessing the activity of the enzyme. We screen kinases from multiple suppliers, and there are big differences. It's amazing. But we'll then identify the most optimized panel that allows you to use, ideally, full-length and small tags. We use physiological substrate sequences, and so that's really important. There's lots of peptide substrates that had been used historically that were modified to get binding to P81 paper. So they have multiple lysines, multiple arginines, and we can take one of those residues off at a time and show that it changes the substrate property. So we're using, historically, these substrates that give you then non-physiological signals. we can use one platform to measure all of these kinases, and we're adding new assays all the time. So here's just a view. So if you use something like staurosporin, right, all the red balls show you the kinases that are inhibited, in this case, greater than 70%. And so it lights up a lot. This is at 1 millimolar. If you do this at 10 micromolar, almost everything lights up. You look on the right, this is a reference compound for EGF receptor, and we have lots of groups sending us reference compounds. And most of these were identified with low ATP assays. They were profiled with low ATP assays, where you see some cross-reactivity, but the idea is that at 1 millimolar, that would disappear, okay? We can measure it at 1 millimolar, and it doesn't disappear. So you see a lot of off-target effects, and this is common across many of the reference compounds, which are, drugs that have been approved. So we have many clients that we're working with, and the level of selectivity that they can achieve when we go back and forth with SAR using our platform is really phenomenal. And so in each case, you're seeing 1 or 2 kinases being inhibited with their lead drugs. And so a lot of the groups that we're working with, their assays, their leads have been fast-tracked through approval by the FDA, both in terms of the cleanness of the assays, but also in terms of the effectiveness of these inhibitors, and really with minimal off-target effects. So, we can work up any one of the hits. We can do IC50s and give you potency. We have many groups sending us covalent compounds. We can do things like doing increasing pre-incubation to understand the rate at which the target is inactivated. So here are 2 EGF receptors, osimertinib and EGF816, and it's amazing how fast osimertinib inactivates EGF receptor, right? And there's quite a shift for 816, so we can tell you right away whether your compounds really are covalent. We can then go on and do Kinact over Ki determinations, which is really what you need to do to analyze the potency of your covalent compounds. And we do this with a statistically validated data analysis program that we've built in ExcelFit. And it allows us then to assess whether it's a 1-step or a 2-step mechanism of inactivation, and to give you then Kinact over Ki, and if appropriate, Ki and Kinact values. And so here's just a whole series of compounds that we've analyzed, where you can see in each one of these circles, they have in essence the same IC50 value, but you can differentiate these based on Kinact over Ki. So that's really important. And we've had clients then send us their cell-based assay data, where it shows an excellent correlation with our Kinact over Ki for their covalent compounds. So this is really important. Another test that we're frequently doing is to look at residence time. And so obviously the longer a drug stays bound to the kinase in a functional manner, right, you can give lower and lower doses. And so this is just from Bob Copeland's paper showing that you can do this by jump dilution, where you pre-incubate your target with 100-fold excess of the IC50 for that particular target. You then do a jump dilution of 100 to 200-fold so that the target cannot rebind the drug that has dissociated. And you just look at the return then of the activity of the enzyme. So on the right, what it shows you is the no drug is the solid circles. A drug that has really fast residence time, so it comes off right away, is going to look like the open circles. So you do the jump dilution, none of it rebinds, so the enzyme is in essence fully active. As the residence time increases, then you get these arcing curves, and if it is covalent, you're going to see a very flat recovery. And in essence, if it's fully irreversible, you won't see recovery at all. So on the left, we can look at a series of EGF receptor mutants with different drugs. We can look and see the untreated is in blue, 2 reversible compounds are in red and yellow, and then 2 irreversible compounds are shown in green and purple at the bottom. So the longer the residence time, right, the slower the recovery. So you expect this for a covalent compound, But on the right is an analysis of 30 different compounds that are all reversible for another tyrosine kinase. And what you see is the vast majority then stack up with the same IC50 value, but their residence time varies from 30 seconds to 2 days. Okay, so really dramatic with a reversible compound based on doing structure function analysis and designing the drugs so that they target the appropriate space for long residence time. Okay, a lot of other things that we can do with the platform. I've talked, mostly about inhibitors, but we see more and more groups coming to us and saying, well, can you use your platform to look at activation? And so if a drug targets a particular target that is downregulated in disease, how do we increase the activity? And this is also where what you have to do is identify physiological sequences that don't inhibit the conformational changes that result in activation. So we can identify sensors that do this very well, shown here for AMPK, where you see about a 3-fold activation with AMP, DNA PK, 30-fold activation with DNA, and so forth. So PKCs with lipids and with calcium, looking at PKR plus or minus RNA, and other CAMKs with plus or minus calcium. We can also then take inhibitors or activators then and look at the mechanism of action. So we could determine then whether it's non-competitive, uncompetitive, or competitive. And as I mentioned, we've really started to invest in making more selective substrates. And so Barbara Imperiali at MIT has pioneered this to show that you absolutely can make selective sensors. In some cases, it's a single peptide sequence. In other cases, you might incorporate a docking domain so that you get then more selective interaction with the target of interest. So one can look at cells and make lysates. One can look at matched tissue samples, which is what she's done in one particular paper that we can provide you. And so in that case, she's looking at, for example, cancer versus normal from the same tissue, from the same individual. You make lysates then of these different samples. Again, you analyze them the same way that I've outlined, and you get then very selective readouts showing selective detection of activity of particular kinases. Or even isoforms. And so we've been able to generate selective sensors for individual MAPK isoforms. We've been able to generate selective sensors for various isoforms of other target families. So very powerful approach that we're starting to work with groups who want to take these assays on into the clinic as a companion biomarker assay so that you can really start to understand which patients should respond, how do they respond to the drug, and if resistance develops, you want to see it at the earliest possible time so you can change the drug cocktail and lead to then more effective management of disease. So last slide, just to show you, there's a lot of talks about Protacs and it's a really exciting approach. One of the things that we've then been able to show is that it's not only important to look by Western blot whether you get knockdown of your target, but also to look by activity, to understand what is the activity of the residual population of enzyme. And so to do this, then what we did was we took MULT4 cells and we treated them or not with a multi-kinase degrader from Nathaniel Gray's lab. And so they were able to show that 90% inhibition of 193 kinases. And so we made then lysates from those treated cells, treated either with control, DMSO, or the specific Protac degrader. And then just do a simple, you make lysates and immediately in the same plate, you can just assay, add the sensor, add the reaction buffer, and be able to detect activity of the individual kinases that sensor would detect. And so in the first example here, the AQT 490 sensor sees the MAP kinases, and you get the expected response. So you see high signals with the control and with the mock degraded sample. With the degraded sample shown in green, you see, a reasonable amount of knockdown. It's not 90%, but it's greatly diminished. However, if we use sensors for CDKs, shown on the right, what you see is actually the opposite response. So the blue and the red then down here is showing you the mock or the controlled DMSO treatment, and the green is actually higher activity. So even though the CDKs are knocked down 90%, the residual 10% has 3 to 4 times the activity. And so this is an important thing to look at so that you understand how the cells have counter-regulated in order to be able to overcome degradation of the target. So just with that, a lot of advantages of the platform. It is sensitive, it can be continuous or endpoint read. It measures the enzyme activity directly using physiologically relevant sequences. You get the complete progress curve without any work. So that is, we typically run 2-hour time courses, and it's just add and read and walk away. Everything is measured under optimal enzyme conditions, both in terms of the sensor as well as with ATP. And we're looking at the linear portion of the progress curve so that you're getting a really accurate measure of activity, which is a reaction rate. Again, accommodate any ATP concentration. We're typically using 1 or 2 millimolar. Because you are measuring a reaction rate, it's very high accuracy and precision with Z primes typically greater than 0.7, and as I showed you, can be greater than 0.9. This platform corrects for any compound autofluorescence. You can do that kinetically because the autofluorescence doesn't change over time, but if it's really severe autofluorescence, then you just use the endpoint red format, and that will eliminate it. So rapid and predictable development. We can develop assays in one week. So we have 30,000 sensors that we've built up for all different branches of the kinome, and we can go in and screen certain subsets and immediately pull out assays. If we follow that milestone process that I outlined, that takes us about 12 weeks to do the whole assay development. Rigorous manufacturing, we see great lot-to-lot consistency with these sensors because they are peptides. And again, works across the entire workflow as shown here. So you can use the PhosphoSens red up front for HDS and SAR, shift over to the kinetic format with the same sensor for mechanism of action, potency assessments, profiling, and then use selective sensors in the PhosphoSens lysate format. And it can be the same sensor for all 3 configurations. So we've offered then now an increasing number of catalog products and services. So we have almost 400 assays for kinases and phosphatases now. We have the custom assay development service that we've outlined, and we have a series of custom testing services, again, all focused on optimized detection of activity to give you the best data for your lead compounds. That includes kinase profiling with 372 targets now, IC50s, Kinact over Ki, residence time, mechanism of action, and EC50s for activation. So with that, thank you so much for your time, your interest, and I would take any questions. And we're also at booth 315 if you want to stop by and meet the team and ask any other questions. Thank you.